This video is based on the work of two research teams. One led by Dr. Owen Standen at the Wellcome Laboratories of Tropical Medicine in the early 50s, and the other led by Dr. Diane McLaren in the late 80s at the National Institute for Medical Research. Dr. McLaren's scanning and transmission electron micrographs of the schistosome are featured in our video. This is the image seen in the microscope, which is enlarged still further when processed photographically. It should be appreciated that these electron micrographs represent a wide range of magnifications. For example, this adult male schistosome is magnified about 35 times when viewed on a 50-centimeter television screen. His ventral sucker, 500 times, and this spiny inside edge of the sucker, 7,000 times. Some of our images are magnified around 250,000 times, like this section showing the outer surface of a larval schistosome. It was with the aid of this electron microscope that Dr. McLaren was able to unravel one of the most puzzling stages in the life cycle of the schistosome. The crucial processes involved in the transformation from free-living organism to a parasitic way of life. These happy children are playing in water that is more than likely to be infected by one of the world's most damaging parasitic diseases. Schistosomes are parasitic flukes that live in the bloodstream of man. They cause the disease Schistosomiasis, probably better known as bilharzia, a major health problem for hundreds of millions of people in many tropical and subtropical regions of the world. A male schistosome is seen here with a female partly held in his gynecophoric canal. The male is about 1 centimeter long and uses the lateral folds of his body to form the gynecophoric canal with which he embraces the longer, more slender female. Incidentally, the scientific name Schistosoma means cleft-bodied. This is the anterior part of the cleft which surrounds the head end of the female. Here are the paired schistosomes in the portal vein of an experimentally infected hamster. The flukes migrate as far as they can into the mesenteric veins, and the female extends out of the gynecophoric canal to deposit her eggs into the smaller blood vessels. The eggs then work through the tissues into the lumen of the intestine. In this species of schistosome, the eggs pass out later with the feces. Here's an egg in a fecal smear. The earliest records of urinary schistosomiasis are to be found in the Ebers Papyrus, dated around 1900 BC. Although the ancient Egyptians did not know the cause of the disease, they recognized and clearly illustrated the urinary symptoms in their hieroglyphics. Calcified schistosome eggs have also been identified in the kidneys of mummies more than 3,000 years old. It was not until 1851 that the German pathologist Theodor Bilharz identified Schistosoma hematobium as the causative agent of urinary schistosomiasis in Egypt. In 1904, Katsurada described the life cycle of Japonicum, and in 1907, Sambon described Mansoni as a separate species. In 1934, Fischer recorded the existence of Intercalatum, and as recently as 1978, Vogue, Bruckner and Bruce discovered Mekongai. Today, 18 species are recognized. Of these, Mansoni, Japonicum and Hematobium probably infect more people worldwide than any of the other species. Mansoni lives chiefly in the veins of the large intestine, and this part of the portal circulation is also infected by Japonicum. The passage of eggs through the intestinal tissues causes ulceration, thickening and fibrosis of the bowel wall. Eggs can be seen in this much thickened region of the superficial layers of the intestine. Also at the bases of the villi. Those eggs which enter the lumen of the gut are discharged with the feces. Schistosoma hematobium differs from the other two species. It inhabits the blood vessels of the bladder and its eggs are discharged with the urine. The main clinical sign is hematuria, but ultimately fibrotic lesions may develop throughout the whole of the urogenital system. Bladder cancer is not an uncommon development in advanced stages of the disease. Schistosomiasis is currently endemic in 75 countries. Hematobium is present in 53 countries, mostly in Africa, but also in the eastern Mediterranean. Mansoni shows a similar distribution, except that it's also found in South America, notably Brazil, where it was probably carried with the slave trade. Japonicum, on the other hand, is confined to countries of the Far East, China, Indonesia, and the Philippines. Japan, where the parasite was originally found, has now been freed from the disease. Mekongai occurs in Southeast Asia. And intercalatum is found in Central and West Africa. Worldwide, some 200 million people are infected with the schistosomes, and 500 to 600 million are at risk. The highest infection rates are found in Brazil, Egypt, and the Sudan. Freshwater snails play an essential role in the transmission of the disease, since each schistosome will spend part of its life cycle in the tissues of a snail. Species of Bulinus are intermediate hosts for hematobium. Biomphilaria carries Mansoni, and Oncomelania carries Japonicum. Wholesale destruction of the snail hosts with chemical molluscicides is one obvious method of reducing the incidence of schistosomiasis. But since the snails are hermaphrodite, possessing both male and female sexual organs, it needs only one individual to survive a control program of this kind for an entire area to be repopulated within a single season. Moreover, development projects such as hydroelectric schemes like the gigantic Kariba Dam on the Zambezi have created large bodies of water and consequently enabled the snails to flourish in recent years. Although most infected people carry only small numbers of schistosomes, treatment with an effective drug not only reduces the prevalence of the disease, it also reduces morbidity. Praziquantel is currently the drug of choice, since a single dose affects all the main species of schistosome that infect man, and it produces high cure rates. Oxamniquin is also used for Mansoni, especially in South America, while metrifonate is valuable for the treatment of hematobium. It's man's primitive habits with regard to urination and defecation which maintain the disease in the community. Obviously, the separation of urine and feces from waters inhabited by the snails is vital in the control of infection. In the Third World, this is easier said than done. However, in Zimbabwe, the Blair research laboratories have produced some interesting designs for simple water pumps and lavatories. This one is a strong shelter built over a deep pit. It has an odor pipe, fly traps, and is easy to keep clean. And here are some variations on the basic design. The Blair water pumps are ingenious. This one uses the pipe that delivers the water as the pump handle. In the playground of a village school, these girls are enjoying their swing pump. Official notices, pamphlets and posters are also used in an attempt to increase local awareness in endemic areas. The most desirable control measure would undoubtedly be a one-shot vaccine, and it's towards this goal that current research is mainly directed. All species of human schistosomes have essentially the same life cycle. We've chosen Mansoni for discussion here, as it's the easiest to maintain and investigate under laboratory conditions. The flukes live paired in the hepatic portal and mesenteric veins. The male is light in colour, with a dark central line, the gut, just visible. The female, partly held in his gynecophoric canal, appears very dark in comparison, because her gut is more obvious. The dark colour is haematin, a product of hemoglobin digestion, derived from red cells of the host's blood, which fills the double zigzag line of the gut. If you look closely, you'll see individual blood cells moving through the narrower passages. This female has an egg in her uterus. Above the egg is the spiraled vitelline duct. The female is cylindrical and has a relatively smooth surface. But the dorsal surface of the male bears many spiny bosses or tubercles that are thought to anchor him in position by catching against the walls of the blood vessels. There are numerous pits between the tubercles, which presumably serve to increase the absorptive surface area, while sensory organelles are abundantly distributed over the entire body. These ensure that the schistosome is aware of minute changes in its microenvironment. Each fluke has two suckers at the anterior end of its body. The ventral sucker is used by the fluke to attach itself to the walls of the blood vessels. The very spiny surface of the sucker ensures a firm grip. The oral sucker is important for feeding and is used to ingest the red blood cells of the host. The orifice is spiny, and its outer margin is well endowed with sense organs. In this electron micrograph, we can see two blood cells inside the oral sucker. Here, the well-anchored pair seem to be traversed by continuous peristaltic waves, which are probably associated with the rate of blood flow. Female schistosomes produce many hundreds of eggs a day, and these are deposited into a venule of the intestinal wall, where they become tightly lodged. The egg shell is covered with hundreds of needle-like spines which abrade the tissue and enable the eggs to work their way across the bowel wall and through the villi. Here are two eggs in tissue at the bases of the intestinal villi. They're making their way towards and into the lumen of the gut. Later, they'll pass out of the body with the feces. Some eggs are inevitably swept back, past the flukes, and carried in the portal blood to the liver, where they eventually provoke an immune reaction and become encapsulated in a granuloma. These granulomas are clearly visible as white spots on the liver of an infected mouse. An egg is at the center of this mass of infiltrating leukocytes, mainly mononuclear cells and eosinophils. These cells ultimately destroy many eggs. They've already started to invade the shell of this one. In severe chronic infections, the liver becomes filled with granulomas, and this leads to portal obstruction, hypertension, ascites, and massive enlargement of the liver and spleen. Compare the size of the organs in a normal mouse and a mouse harboring a chronic schistosome infection. These features of pathology accurately mimic the clinical symptoms of the human disease. Schistosome eggs are easily detected in fecal smears. They're about 1/7 of a millimeter long and are comparatively large as helminth eggs go. The prominent lateral spine is the characteristic feature of Schistosoma mansoni. The spine is terminal in Hematobium. And although lateral in Japonicum, the spine is very small. The Japonicum egg is much rounder and is therefore easily distinguished from the oval-shaped egg of mansoni. When passed in the feces, each egg contains a fully formed miracidium, whose surface is covered with hundreds of cilia. The miracidium also possesses two pairs of flame cells, which can be recognized by their flickering movement. They form part of the simple excretory system, and their activity shows that the organism is alive. In fresh water, the egg expands by osmosis, and as its contents become diluted, the miracidium is stimulated into activity. The movements of its surface cilia set up currents in the liquid, which appears to bubble. Suddenly, the shell is fractured, and the miracidium half emerges together with fluid from the egg. But the miracidium is still contained within the vitelline membrane, which surrounded it in the egg, and it has to struggle hard for a time before it finally manages to free itself. Then, in a moment, it's on its way in search of a particular species of snail. The miracidia are propelled by vibrating the thousands of minute hair-like cilia covering their elongated bodies. This is a critical period for them. They must find their particular species of snail within a few hours, or they'll die. The snail shown here is Biomphalaria glabrata from South America. The miracidia adhere to its exposed surfaces. They burrow their way in by a combination of enzymic digestion and mechanical movement. It takes about an hour for a miracidium to penetrate completely. Once this is accomplished, the miracidium transforms to a primary sporocyst, which migrates towards the liver of the snail and begins a process of asexual multiplication. In this way, many secondary or daughter sporocysts develop independently and grow into long, thin bodies that eventually become convoluted. They give rise to cercariae that bud off in continuous sequence. Finally, the liver tissue of the snail is almost completely replaced by sporocysts and free cercariae. This is a secondary sporocyst which has been dissected out of the liver of an infected snail. It contains mature cercariae which are struggling to get out. They have developed in the sporocyst with their tails folded back along their bodies. This one is struggling very vigorously to free itself. Remember, we are watching cercariae with space around them. If they were still inside the snail, they would be tightly packed and their movements greatly restricted. At about 4 weeks after infection, the first cercariae emerge from the snail, and they're soon escaping daily in swarms. They're about 0.5 millimeter long and are actively swimming organisms that travel through the water with characteristic movements, usually tail first. Their life is short, about 48 hours at most, for they have no functional gut, and their rapidly expended energy reserves cannot be replaced. The cercaria has an elongated body region and a long forked tail. It also has an oral and a ventral sucker by which it adheres to any substrate. The external surface of the cercaria is covered with spines that are backwardly directed. These help it to penetrate the skin of its next host, since they ensure that the organism can only travel in a forward direction. Each cercaria has a secretory complex comprising 3 types of unicellular glands, which open through the oral sucker. This electron micrograph shows the folded gland openings. The glands contain powerful proteolytic enzymes, which help the cercaria to digest its way through the skin of the host. Here, cercariae are penetrating the tail skin of a mouse. They attach with their suckers, digest, and wriggle their way through the skin. Once the body is inside, the tail drops off but remains active. The parasite, now called the schistosomulum, is safely within its new host. The sequence is now repeated in diagram. A cercaria attaches and makes a hole in the epidermis. It deepens the aperture and enters it by elongating its body. Shedding its tail, it migrates through the dermis and fat layer, where it turns, seeking a blood vessel. On finding one, it enters and starts its journey round the body of the host. It's carried in the bloodstream first to the lungs. By this time, it's become longer and considerably more slender, and lost its mid-body spines, although spines are retained at both ends. The spines, together with rhythmic activity, enable it to migrate along the tiny lung capillaries, which are often smaller in diameter than the larva itself. The flukes leave the lungs through the pulmonary veins and pass via the left side of the heart to the liver. The earliest migrants arrive in the liver 6 to 8 days after infection. They become short and squat, begin to feed, and their intestines fill with dark hematine pigment. 2 to 3 week old juvenile flukes are now more recognizable as schistosomes, but they have a convoluted system of folds and ridges on the surface. By week 4, however, these folds are less conspicuous and dome-shaped elevations appear on the dorsal surface of males. They clearly represent early stages in the development of surface tubercles. Male and female flukes pair at around week 5, and the male tubercles take on their characteristic spiny appearance. The paired flukes migrate through the portal vein to the mesenteric veins, and egg-laying begins. That completes the life cycle of the schistosome. We now have some observations on one of the most intriguing features of the life cycle, the transition from free-swimming cercaria to parasitic schistosomulum. The cercaria has to adapt very rapidly to its move from freshwater to body fluid, a temperature change of about 20 degrees, and a hostile environment. The most dramatic changes take place at the outer surface of the cercaria. This is a diagrammatic section through a cercaria. The surface is spiny and totally covered by a typical 3-layered membrane. The outer layer is the glycocalyx. This is an electron micrograph of the 3-layered membrane. Under the membrane is the tegument, a syncytium with no cell walls. Its nuclei are located in deeper cell bodies joined to the tegument by narrow, twisted connections. These are layers of muscle cells. This is how the section appears under the electron microscope. When the cercaria enters the tissues of its new host, large numbers of membranous secretion granules are synthesized in the subtegumental cells and pass up into the tegument. The bodies join together and then connect with the surface of the larva to liberate their membranes to the exterior. The original trilaminate membrane and the glycocalyx are cast off in a mass of microvilli. This action not only serves to distract the host's immune system, it also sensitizes the host against subsequent schistosome infection. The parasite rapidly covers itself with a new 7-layered membrane derived from the membranous bodies. The larva further confuses the host by disguising its surface with proteins derived from the host's red blood cells. At an early stage of infection, it's also thought that the host produces a class of antibodies which attach to the invading parasites. Their function is rather puzzling, in that they're not aggressive towards the parasite. Furthermore, they serve to block the killing of the parasite by other toxic antibodies. Later, there's a switch so that the toxic antibodies predominate. This could explain the finding that young children playing in infected water have no resistance to schistosomiasis and may become heavily infected. But as they get older, they develop an effective immune response that kills newly invading parasites and thus limits the number of schistosomes they carry. Because man eventually develops this partial immunity, concerted efforts are being made in many research laboratories to analyze the mechanisms responsible for killing the flukes and to characterize the parasite antigens against which the host's responses are directed. Present research gives hope that it will be possible to design and produce a vaccine for human use. It would be an acceptable and desirable first step in disease control if the vaccine could reduce the debilitating pathology caused by schistosome eggs. Schistosomes, like many other parasites, have a variety of ways of avoiding the lethal effects of immunity. Shedding of microvilli, using host protein as a disguise, and the development of blocking antibodies. These are but three of their many tricks. A really successful vaccine will have to be several jumps ahead of these highly sophisticated evasive strategies. Until that time comes, we must rely on improved standards of hygiene to break the life cycle of the parasite and drugs to reduce the infection. But the problems of expense, the need for repeated drug administration, and the ever-present threat of drug resistance mean that researchers are working against the clock to develop an effective vaccination program.
These are the paired male and female of one species, Schistosoma mansoni. Several pairs can be seen together here in the portal vein of an experimentally infected hamster. They migrate as far as they can into the mesenteric veins, and the females deposit eggs which pass through the tissues into the lumen of the intestine to be discharged later with the feces. In man, Schistosoma mansoni lives chiefly in the veins of the large intestine, and this part of the portal system is also infected by another species, Schistosoma japonicum. The passage of the eggs causes ulceration, thickening and fibrosis of the bowel wall, and there may also be considerable enlargement of the liver and the spleen. A third species, Schistosoma hematobium, mainly inhabits the veins of the bladder, and its eggs are discharged with the urine. Hematuria results, and ultimately the whole of the urogenital system may be involved. The earliest records of schistosomiasis are found in the Ebers papyrus. The ancient Egyptians didn't know the cause of the disease, but as the hieroglyph shows, they recognized the urinary symptoms. Moreover, calcified eggs have since been demonstrated in the kidneys of mummies more than 3,000 years old. It was 1851 before Bilharz identified Schistosoma hematobium as the cause of urinary schistosomiasis in Egypt. And in 1907, Saint-Bon showed that there was also a second species of adult worm, Schistosoma mansoni. A few years later, Japanese workers described the life cycle of Schistosoma japonicum, and this was followed by Lieper's work in Egypt, which confirmed Saint-Bon's findings experimentally. Today, the Nile Valley is still the chief focus of Schistosoma hematobium, and the incidence there is higher than anywhere else in the world. Schistosoma mansoni exists with it, but occurs chiefly in the delta. Most countries of the Middle East also have a high incidence of Schistosoma hematobium, and it's generally prevalent over the whole of Africa. Schistosoma mansoni is more frequent in the equatorial regions, and it is the only species in South America where it was probably introduced by the African slave trade. Schistosoma japonicum is confined to the Far East, where it appears in China and the Philippines, and to a less extent in Japan. And since it's likely that about 100 million people may be infected, the problems of control and treatment are important. Certain freshwater snails play an essential part in the distribution of the disease, for each fluke must spend a part of its life cycle in the tissues of a particular species. Thus, an Egyptian snail, Bulinus truncatus, is one of the vectors of Schistosoma hematobium. And so is Physoptis africana, which is found in Central and Southern Africa. Australornis glabratus is a vector of Schistosoma mansoni in South America. And Oncomelania hupensis, an amphibious snail from the lower Yangtze Valley, is one of the vectors of Schistosoma japonicum. But while the wholesale destruction of these snail vectors can reduce the incidence of the disease, it is also important to educate the peoples of the endemic areas, and these are some of the official pamphlets and posters which have been issued. For it is not the mere presence of the snails, but rather man's primitive habits which maintain the disease in the community. Such education, however, will take a long time, for it is no easy matter to convince the simple peasant folk in many areas that they must now discard some of their age-old habits. The serious and debilitating effects of the disease are perhaps less difficult to demonstrate, and the treatment of infected individuals is a matter of immediate concern. For this, tartar emetic was first advocated in 1918 by McDonough and Christopherson, and since then other trivalent antimony compounds have been found almost as active, but with rather less toxic side effects. The disadvantage of these antimonials is that they must be injected and are thus inconvenient for use on a large scale. But recently, leucanthone hydrochloride, also called nilodin or myricil D, has been found effective against Schistosoma hematobium when given orally. This is one of a series of compounds first synthesized in Germany in 1938. Its success has stimulated the search for other drugs which may be given orally, and we shall in this film give an account of some of the work which has been done over the past 6 years. For, apart from the synthesis of many hundreds of compounds, it has also been necessary for testing purposes to establish in the laboratory the complete life cycle of the parasite, and this in its turn has led to some experimental investigation of the parasite itself. All 3 human schistosomes have basically the same life cycle, and therefore we shall here discuss one species only, Schistosoma mansoni. This species lives in the veins of the large intestine, where the male and female worms lie paired. The male is light in color, and its body is folded to form the gynecophoric canal in which the female lies partially enclosed. Their relative positions are seen better at a higher magnification. The average length of the males is about 1 centimeter. The females are a little longer and much narrower, and they appear dark in comparison with the males. This is because the worms feed on the red blood cells of the host, and the blind gut, which is larger and more obvious in the female, contains much hematin, a product of hemoglobin digestion. And here too, between the double zigzag line of the gut, can be seen an egg in the uterus, and above it the spiraled vitelline duct. At its anterior end, each worm has 2 suckers, one oral and one ventral, with which to attach itself to the walls of the blood vessel. These are much larger and probably more functional in the male. See how he bends and twists about, searching for a hold. In the experimental animal, the worms are seen to be traversed by continuous peristaltic waves. The purpose of these is not known, but it may be associated with the rate of blood flow. To deposit eggs, the female reaches forwards into the venule of the intestinal wall, where the eggs become tightly lodged. The eggs then work their way through the bowel wall and escape into the lumen of the intestine. They pass out of the body later with the feces. But some always get swept back past the worms, and these are carried in the portal blood to the liver, where they are eventually encapsulated. The eggs may be detected in a fecal smear, which has been prepared from an infected mouse. They are about 1/7 of a millimeter long, and are rather large as helminth eggs go. The prominent lateral spine is the characteristic feature of the egg of Schistosoma mansoni. When passed in the feces, each egg contains a fully formed miracidium with a ciliary coat. The miracidium also possesses two pairs of flame cells, which may be recognized by their flickering movement. One of them is almost in the center of the picture, another is at the top on the left. These flame cells are part of the simple excretory system of the miracidium, and their activity shows that it is alive. In fresh water, the egg expands by osmosis, and as its contents become diluted, the miracidium is stimulated into activity. The movements of its cilia set up currents in the liquid, which appears to bubble and boil. Suddenly, the shell is broken. The miracidium half emerges, and with it much of the contents of the egg. But the miracidium itself is still contained in the vitelline membrane which surrounded it in the egg, and it has to struggle hard for some time before it finally manages to free itself. And then, in a moment, it has gone underway on its search for a particular species of snail, in which it will spend the next stage of its life cycle. The rounded shape of the miracidia becomes elongated as they swim about, and they must find the appropriate snail within a few hours, or they die. The snail shown here is Australorbis glabratus from South America. The miracidia adhere to its exposed soft tissues. One can be seen at the bottom of the picture, already attached to the snail. And they burrow their way in by a combination of digestion and mechanical movement. It takes about an hour for a miracidium to penetrate completely, and once this has happened, it then begins a process of asexual multiplication within the tissues of the snail, a fate which we now follow in diagram. Many miracidia are here approaching the snail. One attacks the exposed surface and penetrates it. Once inside, it is converted to a primary sporocyst. This migrates towards the liver of the snail, and as it does so, many secondary sporocysts are produced inside it. After about the 15th day, the secondary sporocysts develop independently, and they grow into long, thin bodies which eventually become convoluted. Inside them, cercarii are formed, budding off from one end and emerging from the other in continuous sequence preparatory to escaping from the snail. Finally, the liver tissue is almost replaced by sporocyst material and free cercarii. In a secondary sporocyst, which has been dissected out of the liver of an infected snail, the cercarii can be seen moving. These are mature and ready to emerge, and they lie with their tails folded back along their bodies. One is struggling very vigorously to free itself. But it should be remembered that it is seen here under artificial conditions, and that if it were inside the snail, its movements would undoubtedly be much more restricted. Three to 4 weeks after infection, the first cercarii emerge from the snail, and soon they're escaping daily in swarms. They are about 0.5 millimeter in length, and they are actively swimming organisms with long forked tails. They travel through the water with a characteristic movement, usually tail first. Their life is a short one, about 48 hours at most, for they have no functional gut, and their rapidly expended energy cannot be replaced. Each has an oral and a ventral sucker by which it adheres to any surface, and it also secretes powerful proteolytic enzymes which enable it to burrow its way through the skin. The way in which it effects this penetration has been studied by means of sections through a mouse's skin. In the first, the cercaria has become detached, but the broken edges of the epidermis show where the penetration has begun. In the second, its body has elongated and reached into the borehole. Its tail is still attached in this section, and in this one, where it has proceeded still further. But in another, where it is traversing a layer of fat cells, the tail has disappeared. The fifth section shows it entering a lymphatic vessel, and a sixth shows it completely inside one, which indicates that it probably reaches the circulatory system by way of these vessels. This sequence of events is now reconstructed in diagram. The cercaria first makes a hole in the epidermis. It deepens the hole and enters it by elongating itself. It sheds its tail and then balloons itself through a layer of fat cells. Within the layer of subcutaneous connective tissue, it turns and seeks a lymphatic vessel. It finds one and enters it, and so starts on its journey round the body. The schistosomula, as it is now called, traverses the lymphatic system till it reaches the innominate vein. It proceeds to the right side of the heart and thence to the lungs. From there, the route is not known with certainty, but eventually a proportion of all the schistosomulae arrive in the liver. And there, in the intrahepatic portal system, they develop into young male and female worms. These have been extracted from the liver of a mouse about 3 weeks after infection. After another 2 weeks, the worms are sexually mature and pairing begins. The pairs then migrate to the mesenteric veins, where the females start to deposit eggs. And one cycle is complete. For large-scale chemotherapeutic work, mice are the most suitable animals to use, for Schistosoma mansoni infections develop normally in them, and they are small and relatively inexpensive. The rabbit will also take the infection, but though the worms develop to maturity, the eggs are not passed in the feces, and therefore the complete cycle cannot be maintained with these animals. The rhesus monkey, however, takes the infection normally, with eggs appearing in the feces, and the same is true of the golden hamster. It is, however, found best to use the monkey and the hamster for the maintenance of strain only, since they are more expensive than mice and the supply is more irregular. The establishment of a snail colony is an essential part of this work, and a suitable laboratory host is the South American snail, Ostolorus glabratus, for this breeds fairly easily under experimental conditions. It is not difficult to infect, and because of its large size, has a high yield of cercariae. This one is already about 25 millimeters in diameter and will grow even larger. It is entirely aquatic and therefore must be kept in tanks, and it thrives satisfactorily at a temperature of 26 to 28 degrees centigrade. By a careful choice of water plants and animals, the tanks can be established as biologically balanced units so that they require the minimum of attention. The plants maintain the oxygen-carbon dioxide balance, though much gaseous exchange also takes place at the surface of the water. Small crustaceans such as Daphnia, the water flea, eat the free-living protozoa and the unicellular algae which would otherwise foul the water. They are also good indicators of the health of the tanks, for a slight oxygen deficiency quickly causes their numbers to decrease. And at the bottom of each tank is a colony of oligochaete worms. These are tubifex, with their heads buried in the mud and their characteristic waving motion. They eat the snail feces and the decaying vegetation, and thus play an important role as scavengers. The snails themselves also eat the green algae, with which the side of the tank becomes covered, and they are often seen making a stately progress up the tank, eating as they go. The eggs are laid for preference on flat areas near the surface of the water, and the sides of the tank or the broad leaves of the water plant, Ludwigia palustris, provide suitable sites. If sprigs of this plant are floated on the surface of the water, the eggs can be easily collected each day and transferred to the breeding tanks. The young snails soon hatch out. A week is about the usual time at 26 degrees centigrade. In 5 days, they have grown to nearly 2 millimeters in diameter, feeding only on the green algae which covers the sides of the tank. After a week, this food is supplemented artificially. Powdered milk, dried lettuce and grass, ground eggshells, and a vitamin B concentrate are mixed together and suspended in a solution of sodium alginate. This is flooded thinly over the surface of a flat dish. And then calcium chloride solution is poured over it to form the insoluble calcium alginate. Strips of the food thus prepared are given to the young snails, who take it readily and thrive on it. A month after hatching, they are given raw lettuce, and thereafter this forms their main food. At 10 or 12 weeks old, the shells are about 1 centimeter in diameter, and the snails are then at the most suitable stage for infection. For this purpose, myricidia are obtained from eggs present in the feces of infected mice. But the bulk of the fecal material must first be removed, and special care is necessary to prevent the premature hatching of the myricidia during this process. The gut and the liver also contain many viable eggs and may be included if required. A little fine sand is added to facilitate the breaking up of the pellets, and the mixture is gently ground to a paste. Normal saline is added. This solution has a high enough osmotic pressure to prevent the emergence of the myricidia. The mixture is next shaken for 10 minutes to produce an even suspension, after which it is filtered through 2 layers of surgical gauze to remove the coarse debris. These operations are carried out in the cool, for this too will delay hatching. The liquid is then left to settle for a quarter of an hour in the dark. The schistosome eggs sink to the bottom of the flask with the heavier particles, and the lighter particles can be decanted with the liquid. After this process has been repeated twice, the final sediment is washed into a beaker, this time with fresh water, and placed in bright light. In an hour or so, many hundreds of myricidia have emerged, and when the beaker is placed in a beam of light, they become sufficiently visible to be counted as they are removed with a pipette. Meanwhile, the snails are collected from one of the breeding tanks. Experience has taught that excessive handling results in a high mortality rate, so the handling is kept to a minimum by infecting them in batches of 50 instead of singly. 750 myricidia are added to the beaker, that is 15 to each snail. After the snails have been left undisturbed for several hours, while the myricidia penetrate, they are transferred to another tank where they remain whilst the infection matures. And to ensure a high rate of infection, 2 further doses of 5 myricidia per snail are given without removing the snails from the tank. Incubation in the snail takes 3 to 4 weeks, but after this the Cercariae emerge daily when stimulated by warmth and light. And though in some snails the infection disappears after a time, most of them go on producing Cercariae until their death. The mice must be infected individually, but they are handled in batches of 40 at a time. They are first put in a trough containing ½ inch of water so that they will void feces and urine before they're put in their separate pots. This is because the presence of the excreta is thought to reduce the vigor of the Cercariae. These are added from a suspension containing 100 per cc, and 1.3 cc's are withdrawn with a spring-loaded syringe and added to each pot, thus exposing each mouse to 130. The syringe is rinsed after each dose. And in case the fingers are accidentally splashed during this operation, a pad soaked in alcohol is handy. The whole process of infection should be carried out as quickly as possible, for the mice will die if they're exposed to water for very long. When it's completed, the mice are left in the warm for half an hour while the Cercariae penetrate, and then they are dried in sterilized wood wool or straw. "50 days later, the infection should be mature, but this must be confirmed by examining each mouse. A fecal sample is obtained, and from this a smear is prepared, which is inspected for eggs. At autopsy, the mesenteric veins of an infected mouse can be seen to contain many worms. It is only from these worms that the eggs can escape into the feces. A few worms are also present in the portal vein. And the liver and the spleen are both greatly enlarged. The liver is also discolored due to the presence of hematin, and many small abscesses show where eggs have been encapsulated. When the worms are extracted and counted, it is found on an average that 60 to 70% are in the mesenteric veins, 20 to 30% are in the portal vein, and there are a few in the liver. But when the mouse has been treated with an active drug, say tartar emetic, the distribution is quite different. Then no worms at all are found in the mesenteric or portal veins, they are all in the liver. An indication of the activity of a new drug can therefore be obtained from its effect on the worm distribution. This gives a more precise evaluation than merely noting the disappearance of eggs from the feces. 15 mice are used for the test, together with the same number of controls, and the method is demonstrated using nilodin. This drug is given orally, but the interperitoneal injection of a suitable drug would produce the same result. The mice are first lightly anesthetized to facilitate the introduction of a tube into the stomach, and they are given the maximum dose tolerated. The dose is repeated twice daily for 5 days. A week later, the mice are killed and examined. To prevent disturbance of the worm distribution during handling, the portal vein is ligated where it enters the liver. The controls are examined first, and the worms present in the mesenteric veins are picked out and placed in Ringer solution. The males are counted, and the females. Next, the portal vein is excised, and the worms are gently squeezed out. Again, the males and females are counted. Finally, the liver is removed whole and crushed between 2 plates, and it too is examined for worms. Only a few are present, most of them unpaired males. When the treated mice are examined, no worms are to be seen in the mesenteric veins or the portal vein. But the liver and spleen show the characteristic signs of infection. The liver is removed and crushed as before, and it is found to contain many worms, both males and females. The females, however, are not as easy to distinguish as before, for they have become depigmented. The reason for this will shortly be explained. When all the mice have been examined, the results are assembled, and the average distribution of worms in both treated and untreated mice is calculated. From a more detailed study of the worms, it is possible to account for this change in distribution. For in the presence of an active drug, the worm first appears to lose muscle tone. The male then relaxes his hold on the walls of the blood vessel, and the two are swept passively away, back with the portal blood, and into the intrahepatic veins. The dark hematin in the gut of the female disappears, apparently because she has stopped feeding. and both worms become smaller for the same reason. The liver crush from the treated mouse showed the depigmented females, and both males and females here are rather smaller than those in full health. But the worms, though immobilized, are far from being dead, and if treatment is stopped at this stage, many will recover. They can now move away from the liver, back to their former positions in the mesenteric veins. Once more, the eggs appear in the feces, and relapse is complete. To effect a cure, therefore, more prolonged treatment is required. Again, the worms are driven to the liver. But this time, the high concentration of the drug is maintained until they have degenerated so far that they die. The inflammatory tissue normally surrounding the occupied vein increases in extent, and the dead worms are invaded by leukocytes and destroyed. Here is a liver crush from a mouse which has had prolonged treatment. It shows the dead worms already ensheathed in fibrous tissue. In another, the process of destruction has already begun. These effects of relapse and cure can be produced in mice by all the known active drugs. Further, for laboratory purposes, the minimum dose levels producing an initial shift of more than 75% of the female worms to the liver may be used to compare drug activities. On this basis, myricil A, a compound closely related to nilodin or myricil D, has about a third of the activity of nilodin itself, whereas the trivalent antimony compounds, triostam and tartaremetic, are more active than either. Such a comparison provides a means of deciding whether any drug or series of drugs would be worth further investigation. Meanwhile, the search for the ideal drug goes on, and with it the further study of the biology of the schistosomes at all stages of their life cycle, the investigation of the snail vectors, and the most efficient means of controlling them. These and other aspects of the disease are now being studied by organizations all over the world under private or government sponsorship, and each is contributing its quota to our knowledge of this dangerous disease.